Okay, let's dive in. We spent some time looking at the core components of your nervous system, the neurons themselves, right? These amazing cells designed for sending signals fast.
And their support crew, the neurolia, and we talked about the myelin sheath that helps those electrical signals really, zip along the axons.
Exactly. So we've got the wiring, how a signal travels along one nerve cell.
Right. But the real action, the complexity, it happens when one neuron needs to communicate with the next one. How does that message actually get passed along?
That's exactly where we're going today, the absolute next step, the synapse. This is that specialized meeting point where the message handover happens.
And for this discussion, we're leaning on some excellent notes. They're actually lecture notes prepared for nursing students, drawing from that key text, The Human Body in Health and Illness by Barbara Herlihy. Mr. Hostler's notes, basically.
Right? Designed to make these complex ideas accessible. So our mission today is really digging into this tiny, tiny spot. Because honestly, everything your nervous system manages, moving muscles, your mood, making memories, it all hinges on communication across these synapses.
It really does. So we need to unpack the structure and maybe more importantly, the whole process. How does it work?
Let's start with the parts list. The anatomy of a typical chemical synapse. What are the key players?
Okay, so think of three main parts. First, you've got the neuron that's sending the message. We call that the presynaptic neuron, the sender.
Presynaptic, before the synapse.
Yeah.
Got it. The sender.
Exactly. Then on the other side, you have the neuron that's receiving the message. That's the postsynaptic neuron.
Postsynaptic. After the synapse, the receiver. Makes sense.
Right. And crucially, what's between them?
I always pictured them just like touching, like wires.
Ah, see, that's the common misconception, and it's super important here. At the chemical synapse, they don't actually touch. There's a physical space, a tiny gap filled with fluid.
Again.
Yep, called the synaptic cleft. It's incredibly narrow, maybe 20 to 40 nanometers. That's billionths of a meter, but it's a real gap.
Wow. Okay, so if there's a physical gap, that electrical signal speeding down the axon of the first neuron,
Yeah.
it can't just jump across that space.
Precisely. It hits a dead end, electrically speaking. And that's why the communication across this cleft has to be chemical, not directly electrical. The signal needs to change its form.
Okay, so it needs a chemical messenger.
It does. And this rupture sender gap receiver also means the communication only goes one way, from presynaptic across the cleft to postsynaptic. It's unidirectional.
That makes sense. You can't send a message backwards across that setup.
Huh.
So how does this electrical signal arriving at the end of the sender neuron, how does it trigger the release of this chemical messenger? Walk us through that sequence.
Okay, step by step. First, that electrical signal, the action potential we talked about before, it travels all the way down the axon of the presynaptic neuron.
Right to the very end, the axon terminal.
Exactly. When that wave of electrical change, the depolarization, reaches the terminal, it causes special doors to open in the membrane there. These are voltage-gated calcium channels.
Calcium channels. Okay, so voltage-gated means the electrical signal opens them.
Yep. And when they open, calcium ions, which are more concentrated outside the neuron, rush into the presynaptic terminal.
So calcium floods in. What does the calcium do?
That influx of calcium is the key trigger. It's the signal inside the terminal that says, okay, time to send the message. It causes these little membrane bubbles inside the terminal called synaptic vesicles to start moving.
Vesicles.
Yeah.
Like tiny storage bags.
Pretty much. And they're filled with the chemical messengers, the neurotransmitters. The calcium causes these vesicles to move towards the edge of the presynaptic membrane, right up against the synaptic cleft.
Okay.
And then they fuse with that membrane. Imagine them merging with the outer wall and basically dumping their contents out into the synaptic cleft.
Ah, okay. So they release the neurotransmitters into that gap.
Precisely. So the electrical signal arriving at the terminal gets converted into this release of chemicals into the cleft. That's the crucial transformation.
Electrical to chemical. Now, these neurotransmitter molecules are just floating in that tiny fluid space.
They are. And they just drift or diffuse across that very short distance. It doesn't take long.
And the postsynaptic neuron, the receiver, it needs a way to catch that message, right?
Absolutely. Embedded in the membrane of the postsynaptic neuron are specific receptor proteins. Think of them like perfectly shaped docking stations or locks.
And the neurotransmitters are the keys.
Exactly. Each type of neurotransmitter molecule has a specific shape, and it will only bind to receptors that have the complementary shape. It's highly specific.
So the right key fits into the right lock. And when that binding happens, what's the effect on the receiving neuron?
Well, that binding event triggers a change in the postsynaptic neuron. Depending on the specific neurotransmitter and the specific receptor it binds to, it can either make the postsynaptic neuron more likely to fire its own electrical signal, its own action potential.
That sounds like excitation, like a go signal.
Exactly, that's excitation. Or the binding can make the postsynaptic neuron less likely to fire an action potential.
Okay, so inhibition, a stop or slow down signal.
Right. So let's recap that flow. Electrical signal down the axon, hits the terminal, opens calcium channels, calcium rushes in, triggers vesicles to fuse, release chemical neurotransmitter into the cleft, neurotransmitter drifts across, binds to specific receptor on the postsynaptic neuron.
And that binding causes either excitation or inhibition in the receiving cell, potentially triggering a new electrical signal if it's strong enough excitation.
Wow.
Electrical to chemical, then back to electrical potentially.
That's the essence of it. And this conversion point, the synapse, it's not just a simple relay switch. It's a point of integration, of modulation. The postsynaptic neuron might be receiving signals from hundreds or thousands of other synapses at the same time, some excitatory, some inhibitory.
Ah, so it's adding up all those signals to decide whether it should fire.
Exactly. It's integrating all that input. That's fundamental to how your brain processes information. And it's also why this tiny junction is so incredibly important clinically.
Right, you mentioned this is where things can go wrong in diseases and where drugs often work.
Absolutely. So many neurological conditions involve problems with synaptic transmission, and countless medications from anesthetics to antidepressants to muscle relaxants target specific steps in the synaptic process.
It really drives home why understanding the details here matters. Now, neurotransmitters, the chemical messengers. You mentioned acetylcholine earlier.
Yes, allyl-choline, often abbreviated ACEC. It's a really key one, especially famous for being the neurotransmitter used at the neuromuscular junction. That's the synapse where a motor neuron tells a skeletal muscle fiber to contract.
So basically telling your muscles to move. Is it excitatory there?
Yes, at the skeletal neuromuscular junction, AC is excitatory. It makes the muscle cell membrane depolarize, leading to contraction.
Okay. What are some other major players mentioned in the notes, things we might hear about more often regarding the brain?
Well, there's dopamine, hugely important for things like voluntary movement. Think Parkinson's disease, where dopamine-producing cells are lost, but also for motivation, reward, and mood.
Right, the feel-good neurotransmitter sometimes.
It's involved in that, the brain's reward pathways. Then there's serotonin, also heavily involved in regulating mood, but also appetite, sleep, and aggression. Widely known because of its connection to depression and anxiety treatments.
Okay, dopamine, serotonin. What about those excitatory and inhibitory ones you mentioned?
Ah, yes, crucial for balance. The main inhibitory neurotransmitter in the brain is GABA, that's gamma-aminobutyric acid. Its primary role is to reduce neuronal excitability throughout the nervous system. Think of it as applying the brakes, calming things down.
GABA, the main brake. Got it. And the main accelerator.
That would be glutamate. It's the most abundant excitatory neurotransmitter in the brain. Its job is generally to ramp things up, make neurons more likely to fire. It's critical for learning and memory, actually.
So you've got glutamate saying go and GABA saying whoa, and the brain is constantly managing this balance.
Constantly. Every neuron is listening to a chorus of excitatory and inhibitory inputs from all the synapses connecting to it. The overall function of your nervous system depends critically on maintaining the right balance between these opposing signals in the right circuits at the right times. Too much excitation can lead to seizures, for example, while too much inhibition can cause sedation or unresponsiveness.
That makes perfect sense. Okay, so the neurotransmitter delivers its message excite or inhibit by binding to the receptor. But then what? It can't just stay stuck there forever, can it? That would lead to constant signaling.
Absolutely not. That would be disastrous. The signal needs to be brief and precisely timed. So once the message is delivered, the neurotransmitter has to be cleared out of the synaptic cleft very quickly. This allows the postsynaptic neuron to repolarize or reset, ready for the next signal, and prevents overstimulation.
So how does the body clean up that synaptic cleft? What are the mechanisms?
The notes lay out three main ways this happens. First, there's reuptake. The presynaptic neuron, the one that released the neurotransmitter, often has transporter proteins on its surface that act like little vacuum cleaners. They actively grab the neurotransmitter molecules from the cleft and pull them back inside the presynaptic terminal.
Oh, recycling, so it can be repackaged into vesicles and used again.
Exactly. It's very efficient. That's reuptake. The second mechanism is enzymatic degradation. There are specific enzymes located right in the synaptic cleft or sometimes on the postsynaptic membrane, whose job is to break down the neurotransmitter chemically.
Like little Pac-Man chewing them up.
Kind of, yeah. A classic example is the enzyme acetylcholinesterase, which specifically breaks down acetylcholine in the neuromuscular junction and other synapses. It chops it up very rapidly, inactivating it.
Okay, so reuptake or getting broken down by enzymes. What's the third way?
The third way is simply diffusion. The neurotransmitter molecules can just drift away from the high concentration in the cleft out into the surrounding extracellular fluid where they're too dilute to have significant effect on the receptors anymore.
So vacuumed back up, chopped up by enzymes, or just floating away. Those are the cleanup options.
Those are the main three, yes. Reuptake, enzymatic degradation, and diffusion. They ensure the signal is transient and the synapse is ready for the next round of communication.
And thinking back to the clinical relevance, you mentioned drugs targeting these processes. Reuptake seems like a big one.
It really is. Blocking reuptake is a common strategy. We mentioned antidepressants earlier, SSRIs, selective serotonin reuptake inhibitors. What do?
They inhibit the reuptake of serotonin.
Right. They block those vacuum cleaner transporters on the presynaptic neuron that normally pull serotonin back in.
So more serotonin stays in the synaptic cleft for longer.
Exactly. It hangs around in the gap longer, giving it more opportunity to bind to the postsynaptic receptors and enhance serotonergic signaling. The idea is that this might help correct imbalances thought to contribute to depression or anxiety.
Wow, that directly links understanding this cleanup mechanism to how a major class of drugs works. It really makes you see the synapse as this dynamic, controllable point.
It absolutely is. Okay, so let's quickly recap the key points about this chemical synapse.
Right. It's the communication junction between neurons.
Involves a sender, presynaptic, a receiver, postsynaptic, and that crucial fluid-filled gap, the synaptic cleft.
Communication across the gap is chemical, not electrical.
Right. An electrical signal triggers the release of chemical messengers, neurotransmitters from the presynaptic side.
Those neurotransmitters diffuse across the cleft and bind to specific receptors on the postsynaptic side.
Triggering either an excitatory or inhibitory effect in the receiving neuron.
And the signal flow is one way only, presynaptic to postsynaptic.
And finally, critically, the neurotransmitter must be quickly cleared from the cleft to terminate the signal and reset the synapse.
By reuptake, enzymatic breakdown, or diffusion.
That's the essence of chemical synaptic transmission, an incredibly elegant and fundamental process.
Billions and billions of these tiny events happening constantly, underpinning everything. Okay, so building on that, especially the cleanup part, here's something to think about, drawing from the notes approach. Imagine a substance, maybe a medication, or even a toxin, that works by blocking the enzyme acetylcholinesterase, the enzyme that normally breaks down acetylcholine at the neuromuscular junction.
Okay, so the enzyme that cleans up ACA is inhibited.
Right. What would you predict the immediate effect of that substance would be on muscle activity? Think about ACA staying in the synapse longer than it should. What happens to the muscle?
Interesting question. It forces you to apply the mechanism directly. What happens if the off switch is jammed?
Exactly. Something to mull over. And understanding this detail, this synaptic mechanism, it really sets the stage for where we can go next. We've got the neuron, we've got the synapse. Now we can start zooming out.
Looking at the bigger picture, how these neurons and synapses are organized.
Precisely. Starting to explore the major regions of the brain itself. Looking at how the structure of different brain areas relates to their specific functions, building up from the micro-level communication to the macro-level organization.
It's a logical progression, from the basic units to the complex structures they build.
Absolutely. Well, thanks for joining us on this deep dive into the synapse. Keep exploring.